Hello everyone. In this video, we will discuss brief answer question asked in MUHS Supplementary 2023 Paper 1. So let's begin. These were the all brief answer question which were asked. Let's discuss each of the question in detail. First question is, explain with reasoning what is good cholesterol and bad cholesterol. So this is brief answer question of 2 mark. So 1 mark each is allotted for writing answers to good cholesterol and bad cholesterol. Okay. So what is the answer? Which is the good cholesterol? HDL cholesterol, that is high density lipoprotein cholesterol, is termed as good cholesterol. Now for reasoning, you have to, for if you write the reason, you get another half mark. So because it collects excess cholesterol from tissues and transport it to liver where it is excreted in the bile. Hence, HDL cholesterol is considered as anti-atherogenic and cardioprotective. Okay, so this is for 1 mark, as opposed to LDL cholesterol or low density lipoprotein cholesterol is termed as bad cholesterol because it transports cholesterol from liver to peripheral tissue. Hence, elevated level of LDL will cause atherosclerosis and which may lead to myocardial infarction. So that is why it is called as bad cholesterol. So HDL cholesterol is good cholesterol because cholesterol is transported from tissue to liver. Okay, this transport is called as reverse cholesterol transport. Okay, and LDL cholesterol is termed as bad cholesterol because cholesterol is transferred from liver to peripheral tissue that can cause atherosclerosis and which may lead to myocardial infarction. Okay, so if you write this, that is, you will get 2 out of 2 marks. Okay, now let's see the next question. Why copper deficiency lead to defective collagen formation? Okay, now here you have to remember your functions of copper and which enzyme contain copper. Okay, and what is the role of that enzyme in collagen formation? Okay, so what is the enzyme which contain copper which has role in collagen formation? That is lysyl oxidase. Okay, lysyl oxidase is an enzyme which contains copper, and this enzyme is required for the formation of covalent cross linkage in the collagen. Okay, this enzyme is required for covalent cross linkage. This word is very important. Okay, don't write directly that formation of mature collagen. How it is mature? We have to produce cross linkage in collagen. Now after writing this, mention the reaction which is catalyzed by lysyl oxidase. Okay, which reaction is catalyzed? So there are lysine residue in the collagen structure. Okay, lysyl oxidase converts lysyl residues to L-lysine residue. Okay, lysine residue is converted to L-lysine residue. It is by oxidative deamination. Okay, by oxidative deamination, lysine residue of the collagen molecule is converted to L-lysine residue. In the next step, this L-lysine residue will condense with the another lysine residue on the neighboring collagen molecule. Okay, L-lysine will condense with the lysine residue on the neighboring collagen molecule, and this will lead to the formation of crosslink. And after the formation of crosslink, there will be the formation of mature collagen. Okay, there will be the formation of mature collagen. So that is why copper deficiency lead to defective collagen formation. Okay, so you have to recollect what you read during the mineral part. Okay, which enzyme contain copper and what is the role of that enzyme in the collagen formation. So enzyme is lysyl oxidase. Okay, what is the role of lysyl oxidase? You have to write conversion of lysine residue in collagen to L-lysine residue. In the next step, you have to also mention how cross linkage are produced. Okay, so L-lysine residue will condense with the lysine residue on the neighboring collagen molecule. Okay, now this will lead to the formation of cross link. Okay, and then mature collagen fiber will be produced. So if there is a deficiency of copper, there will be the defective collagen formation. There will be no cross link and immature collagen fiber will be produced. So that is why copper deficiency lead to defective collagen formation. Okay, let's see the next question. Define specific dynamic action of food, that is SDA of food. What is SDA of protein? So 1 mark will be for writing the definition, okay, and 1 mark for writing SDA of the protein. So let's see the definition. What is SDA or specific dynamic action? Amount of energy expenditure above the resting metabolic rate, okay, due to the ingestion and digestion of food for use as energy or conversion to a storage form is called as specific dynamic action of foods. So what it means? Energy expenditure, amount of energy expenditure above the resting metabolic rate. Now what is resting metabolic rate? Resting metabolic rate is a energy expenditure for resting stage, which is used in resting stage, that is post absorptive stage. For example, when you are during respiration or during ion transport or flow of blood, so whatever the energy is required for that energy is called as resting metabolic rate. Okay, now the energy above the resting metabolic rate, okay, energy, amount of energy expenditure above the resting metabolic rate, how it is obtained? By ingestion and digestion of food, and where it is utilized, used as a energy, or it can be converted to a storage form. Okay, so what is HDA of food? Amount of energy expenditure above the resting metabolic rate. Resting metabolic rate is energy expenditure which is used for in the resting stage, that is for respiration, blood flow or ion transport. Okay, so energy expenditure above the resting metabolic rate, due to how it is obtained, due to ingestion and digestion of food. Okay, it can be used as a energy or it can get converted to storage form. This is called specific dynamic action of food. Okay, so this is word is very important, above the resting metabolic rate. Okay, this is called as specific dynamic action. So 1 mark for writing definition. And what is HD of protein? It is 30%. HD of protein is 30%. Okay, so 1 mark for writing 30%. Now, what are will what you should know? What are the HDA of carbohydrate and lipid? So HDA of carbohydrate is 5%, HDA of lipid is 15%. Okay, for your understanding. Okay, it can be asked in a subsequent exam. So if the protein SD is asked in next exam or subsequent exam, SD of carbohydrate and lipid can be asked. Okay, so very easy. Write the definition, 1 mark. Write the SD of protein, 1 mark. Okay, next question. Explain how vitamin E is a lipotropic factor. Now this is very important because most of the books mention the names of lipotropic factor. Okay, choline, betaine, then vitamin E. How vitamin E is a lipotropic factor? Due to its antioxidant property. Okay, that is the only word which is mentioned in most of the book, but that will be not sufficient for writing the answer for 2 marks. So what you have to write? There are two mechanisms by which vitamin E acts as a lipotropic factor. Okay, so to write the answer of this, you should first mention what is lipotropic factor, what is the action of vitamin E in by which it acts as a lipotropic factor. So first point should be what is lipotropic factor. Lipotropic factor, what is it? It is a compound that increases the removal. Okay, compound that increases the removal or decreases the deposition of fat. Okay, increase the removal of fat or decreases deposition of fat in the liver. How? Through their interaction with fat metabolism. So any factor or any compound which increases the removal of fat or decreases the deposition of fat in the liver, that is called as lipotropic factor. Now, how vitamin E is a lipotropic factor, whether it is increasing the removal or decreasing the deposition of fat. What is the role of vitamin E? It decreases the level of membrane transport, a very important, not only due to its antioxidant property, it acts as a lipotropic factor. There is an important mechanism is membrane inhibition of membrane transporter. Okay, decreases the level of membrane transporter. Which membrane transporter? Which is responsible for uptake of fatty acid into the liver. So uptake of entry of fatty acid in the liver is decreased. Okay, this membrane transporter is called as hepatic cluster of differentiation 36, CD36, or it is okay if you remember, it is also okay if you don't remember. Okay, CD36. Okay, cluster of differentiation 36, name of the protein. Membrane transporter is nothing but a protein. So it decreases the level of membrane transporter, which is responsible for the uptake of fatty acid to the liver. So entry of fatty acid to liver is decreased. Okay, so it decreases the deposition of fat in the liver. Okay, now this is the first mechanism. What is the second mechanism? Already known, well known action of vitamin E is antioxidant. Okay, so it is a most potent biological chain breaking antioxidant. Very important, each word is important. Most potent biological chain breaking antioxidant. Okay, now what is the how it act, what is the role of this oxidative stress in causation of fatty liver? Okay, now what is the role of oxidative stress? It plays a central role in the development of fatty liver. Okay, oxidative stress plays a central role, major role is played by oxidative stress for the development of fatty liver. Okay, and vitamin E is an antioxidant, very important, and antioxidant. So, by targeting oxidative stress component, vitamin E prevents development of fatty liver. So, two mechanisms we have seen. Which are the two mechanisms? By virtue of its antioxidant property and decreasing the synthesis of membrane transport. Okay, so this is how vitamin E acts as a lipotropic factor. First, decrease the membrane transporter that transport fatty acid into the liver. Second one is a well-known function of vitamin E is antioxidant property. So due to antioxidant property, it prevents development of fatty acid, fatty liver, because oxidative stress plays a major role in the development of fatty liver. Okay, so these two property you have to write. Okay, now even if you are right only it acts as a lipotropic factor due to its antioxidant property, that will not be sufficient, because you have to then write what is the role of oxidative stress in development of fatty liver. So you have to mention that oxidative stress plays an important role for the development of fatty liver, and this oxidative stress is decreased or inhibited by vitamin E due to its antioxidant property. But that will be only for 1 mark. For 2 mark, you have to write inhibition or decrease synthesis of membrane transporter, okay, which transport fatty acid into the liver. Okay, so these are the two mechanisms by which vitamin E acts as lipotropic factor. Okay, next question. Give two examples of competitive inhibitor. Very straightforward question. Okay, just collect your knowledge from your enzyme part, okay, and one from your metabolism part. Okay, because two examples are asked. Okay, so I have provided 4 examples. So whenever you are reading enzyme chapter in any book, this is a classic example already most of the book give, that is inhibition of malonate, inhibition of succinate dehydrogenase by malonate. Okay, so but when you are writing the examples of competitive inhibitor, this is very important topic for SAQ as well as LAQ. Okay, so when you are writing about inhibition, you should mention the substrate also. Okay, because competitive inhibition means inhibitor is a substrate analog. Okay, it is a substrate analog of, so you have to mention substrate name also. So malonate, okay, substrate is succinate, and enzyme, which enzyme is inhibited? Succinate dehydrogenase. Okay, next one is allopurinol, that is used in the treatment of gout. Okay, this inhibition is called as suicidal inhibition. So allopurinol inhibits xanthine oxidase, substrate is hypoxanthine. Next is statin class of drugs, which is used to decrease the level of cholesterol, which is used to decrease cholesterol level. So statin example is lovastatin. What is the enzyme? Is HMG-CoA reductase. It inhibits HMG-CoA reductase. Okay, substrate is HMG-CoA. Substrate is HMG-CoA. Another example, ethanol inhibits alcohol dehydrogenase, which is used therapeutically in methanol poisoning. Okay, alcohol or ethanol is used in the treatment of methanol poisoning, because these are substrate analog, and ethanol will inhibit alcohol dehydrogenase. So these are the examples of competitive inhibitor. These every book mention this example. First example, that is malonate. Okay, when you are studying metabolism, Then allopurinol, for use for the treatment of gout. So these two will suffice when you are asked about the only two examples. To add, you already must have gone through the anti or hypolipidemic drugs. So one drug is lovastatin, okay, or statin type of drug that inhibit HMG CoA reductase. Okay. Then what is the another use of competitive inhibitor? Ethanol is used in methanol poisoning. Ethanol is used in methanol poisoning because it inhibits alcohol dehydrogenase. Okay, so these are the four examples of competitive inhibitor. Next question is name two antioxidants which are used as therapeutic agents. Okay, so which are the antioxidants which have therapeutic uses? First is vitamin A. You know it is an antioxidant vitamin. Where it is utilized, it is used in the treatment of acne. It is used in the prevention of cataract, prevention of cancer. It is also used in HIV/AIDS patients. Okay. Next is vitamin C. Vitamin C is used for the treatment of respiratory infection. Okay, it also have immunological properties. So during COVID, it was also used. Okay, you must have heard the name of the tablet, that is Lim-C, that contains vitamin C. So it was also used in COVID. Okay, so it also has role in wound healing. Vitamin C is also used for wound healing. Next is vitamin E. Just we have seen it is a lipotropic factor, so it prevents the development of atherosclerosis. Okay, it is also used in the treatment of Alzheimer disease. Also used in the treatment of Alzheimer disease. Then comes your antioxidant like copper. Now copper is present in superoxide dismutase enzyme. Okay, it is present in superoxide dismutase enzyme, and this copper is utilized in the treatment of cancer due to its cytotoxic effect. Okay, it is used in the treatment of cancer by virtue of its cytotoxic effect. Okay, next is zinc. Now zinc has a property to increase the osteoblast formation and decreases the osteoclast formation. So it is used for bone regeneration. It is used for bone regeneration, and also it is utilized for wound healing. Okay, also utilized for wound healing because it is required for zinc dependent metalloproteinases. Okay, it is used for a zinc dependent metallic proteinases. Okay, next antioxidant is selenium. Selenium is present in glutathione peroxidase. Okay, and it is also used for the treatment of the cancer or to prevent the development of cancers. Okay, so these are the antioxidants which are used as a therapeutic agent. Most of you must be aware of all these antioxidants. Okay, all these are antioxidants. You all know which are the antioxidant. Vitamin S, how to remember? ACE. These are the antioxidant vitamins. ACE vitamin. Okay. So all they are utilized as therapeutic agent. Okay. In the form of drugs. Okay. Then there are your minerals which are antioxidant properties. So these are also used therapeutically. Okay. So even if you don't remember or get confused due to this question, that is which are used as therapeutic agent, but you know only name of the antioxidant. Okay, but these are used therapeutically as well. Okay, so vitamin A, vitamin C, vitamin E, copper, zinc and selenium. So these are all antioxidants which are used as therapeutic agents. Okay, let's see the next question. So next question is, give biochemical mechanism of cataract formation in diabetes mellitus. Okay. So here I will suggest you to draw a flow chart rather than writing a descriptive answer to this question. Okay. Now I will describe you first the biochemical mechanism or biochemical reactions which are occurring normally, and then what happens in diabetes mellitus. So in normal condition, glucose is converted to glucose 6-phosphate with the help of hexokinase enzyme which requires ATP, and this glucose 6-phosphate is channeled to glycolysis, that is anaerobic glycolysis. So in most of the condition or most of the glucose molecule is channeled for anaerobic glycolysis. Okay, most of the glucose molecule are channeled to glycolysis, okay, and some glucose molecule which do not get phosphorylated follow a different pathway that is called polyol pathway. Okay, that is called polyol pathway. Only 4% of total glucose molecule normally follow polyol pathway. What is polyol pathway? Glucose with the help of aldose reductase is converted to sorbitol, which requires NADPH. Okay, in next step, sorbitol is converted to fructose with the help of sorbitol dehydrogenase. It requires NAD, and then fructose to fructose 6 phosphate. Again, it will channel for glycolysis. Now, this is the normal mechanism which is occurring. Okay, normally most of the glucose molecule is channeled for glycolysis, and only 4% of total glucose molecule inside the lens follow a polyol pathway where it is converted to sorbitol and then fructose. Okay, now what happens? This is normal. Now what happens in diabetes mellitus? Now in diabetes mellitus, the glucose concentration is tremendously increased. Okay, glucose concentration is increased. Now what happens due to increased glucose concentration normally? Now normally also glucose is converted to glucose 6 phosphate. So increased glucose means increased glucose 6 phosphate. Okay, glucose 6 phosphate will increase. But glucose 6 phosphate is an inhibitor of hexokinase. Hexokinase is inhibited by glucose 6 phosphate. Okay, hexokinase is inhibited by glucose 6 phosphate. Okay, so that is why there will be no or limited conversion of glucose to glucose 6 phosphate. So due to this, glucose concentration will increase. Okay, glucose concentration will increase because conversion of glucose to glucose 6 phosphate is stopped. Okay, now due to increased glucose concentration, where it will get channelized to polyol pathway. It will get channelized to polyol pathway where it will get converted to sorbitol. So more amount of glucose molecule means more amount of sorbitol. Okay, more amount of sorbitol pathway. Now what is the more amount of sorbitol? Now what is the next step? Conversion of sorbitol to fructose by the help of sorbitol dehydrogenase. But the quantity of sorbitol dehydrogenase inside the lens is limited. Quantity of sorbitol dehydrogenase inside the lens is limited. Secondly, sorbitol has very low affinity for sorbitol dehydrogenase. Okay, so there will be the less conversion of sorbitol to fructose. Okay, very less conversion of sorbitol to fructose. Okay, so due to this also sorbitol concentration will increase. Okay, that is one. Another important point, there is a limited permeability or low permeability of sorbitol to the lens. It cannot move easily outside the lens. So that's why sorbitol concentration will increase. Okay, more amount of sorbitol will be there. Okay, so what happens? Increased glucose to will lead to increased glucose 6 phosphate. Increased glucose 6 phosphate will inhibit hexokinase. So as the hexokinase is inhibited, glucose concentration will increase. So it will get channeled to sorbitol pathway or polyol pathway where it will convert it to sorbitol. Sorbitol will convert to fructose, but concentration of sorbitol dehydrogenase inside the lens is very limited. Also, affinity of sorbitol to sorbitol dehydrogenase is less. So that's why conversion of sorbitol to fructose will be limited. So that will lead to increased sorbitol concentration. Another important point is sorbitol is less permeable to the lens. Okay, permeability of sorbitol is very less, so it cannot move easily outside the lens. So due to this, there will be the increased sorbitol concentration. So both factor, increased sorbitol and decreased fructose concentration, what it will lead to? It will lead to hyperosmotic effect. It will lead to hyperosmotic effect. That is, increased sorbitol and decreased fructose concentration will cause hyperosmotic effect. Due to this, to balance the osmolality, fluid will move inside the lens. To maintain the osmotic balance, fluid will move inside the lens. Movement of fluid inside the lens. It will lead to liquefaction and swelling of the lens and ultimately the opacity of the lens fiber. Okay, liquefaction, swelling of the lens fiber, and then opacity and you will have the cataract. Then there will be the development of cataract. Now this is the biochemical mechanism for cataract formation in diabetes mellitus. Okay, so I will revise it. Normal, what happens normally? What happens normally? Glucose is channeled to glycolysis. Only 4% of total glucose is follow polyol pathway. But in diabetes mellitus, increased glucose concentration, increased glucose 6-phosphate. But increased glucose 6-phosphate will inhibit hexokinase. So again, glucose concentration increases. It follow polyol pathway. Polyol pathway, first product is sorbitol, second product is fructose, but concentration of sorbitol dehydrogenase is very less inside the lens, so that will lead to increased sorbitol and decreased fructose. Both these will lead to hyperosmotic effect. This will lead to hyperosmotic effect. Due to hyperosmotic effect, fluid will move inside the lens, that will cause liquefaction and swelling of the lens fiber, ultimately opacity and development of cataract. Now this is the biochemical mechanism of cataract formation in diabetes mellitus. Okay, let's see the next question. What is opsonization effect? Okay, this is from immunity chapter. What is opsonization effect? So opsonization is a process of marking the foreign antigen for an immune response by using molecules known as opsonins to eliminate them by phagocytosis. So basically what is opsonization? The marking of the foreign antigen. This is a process of marking the foreign antigen for immune response. Okay. How they are marked? By using the molecules which are called opsonins. Okay, and once the foreign antigen or pathogen is marked by opsonin, then that indicate that foreign antigen is going for phagocytosis. So which are the opsonins? It can be antibodies, for example IgG. It can be complement component like C3b, C4b. It can be proteins in circulation like collectins. Okay, these are opsonin, and opsonization means marking the foreign antigen by using the molecules which are called opsonins. Okay, so what is opsonization? Marking foreign antigen by antibodies, by complement component, and some protein like collectin. Okay. Now we will see what happens during opsonization. It is mediated by Fc receptors which is present on phagocytic cell. Okay, which is present on phagocytic cell, and it binds to the constant region or Fc region of the immunoglobulin, mainly IgG. Okay, so opsonization. So we have seen which are the opsonins: antibodies, complement components, and collectin. For antibody, the opsonization is mediated by Fc receptor which are present on phagocytic cells which bind to this Fc region or constant region of immunoglobulin. Similarly, for complement component, there will be the again receptors. Okay, which are the receptor? Again, these are called as complement receptor. So binding of complement receptor on the phagocytic cell to complement components. Okay, so IgG receptor which bind Fc receptor binds to the Fc component of the antibody, mainly IgG, and binding of complement receptor on phagocytic cell to complement components. Okay, so due to this binding, what happens? Binding of antibody, binding of complement component, binding of proteins like collectin. Due to this binding, there is an activation of signal transduction that facilitates phagocytosis. Okay. After phagocytosis, pathogens are destroyed through enzymatic digestion, that is formation of phagolysosome, or by oxidative damage. Okay. Now this is a foreign antigen. Okay. And what is opsonization? The marking. Okay. Marking by different opsonin. Okay. These are which are the different opsonins. This is antibody, then complement receptor, or sorry, complement components, then some proteins. So this is opsonization. Okay, it binds to specific receptor. Okay, Fc receptor on phagocytosis or on phagocytes, or there are complement receptor on phagocytes. Okay, so there is a invagination and then phagosome formation and phagolysosome formation. Okay, so this is opsonization effect. So basically it is a Marking of foreign antigen by opsonin. Once they are marked, foreign antigen are marked or marked by opsonins, then they are going to be destroyed, okay, with the help of phagocytosis. Okay, this is opsonization effect. Okay, so here you can write what is opsonization effect. Marking of foreign antigen or foreign antigen by opsonin. Opsonin can be antibody, complement, or proteins like collectin. Okay, then there is a binding of receptor of opsonin to the receptor of phagocytic cell, so that will cause invagination. Okay, then there is a formation of phagosome and then phagolysosome. Okay, so there will be the pathogens will be destroyed by enzymatic digestion or oxidative damage. Okay, so this is opsonization effect. Let's see the next question. Next question is, give 4 examples of anti-mutagens. So before that, what are anti-mutagens? Anti-mutagens are agents which decreases the number of spontaneous or induced mutation that will otherwise be caused by mutagens. Okay, agents that decreases the number of spontaneous or induced mutation that will otherwise be caused by mutagens. Okay, so they counteract anti-mutagens counteract the effect of mutagens. What are mutagens then? Mutagens are any substances which have capacity to produce mutation. Mutation can be caused by any physical agent, chemical agent, or if there is an error in DNA replication, DNA repair, DNA recombination. Okay, so that will cause mutation, and ultimately that can lead to cancer or any other genetic disorder. So what are the examples of anti-mutagens? Okay, so it is not given in books, but there is no choice rather than mugging them. So these are cysteine, lipoic acid, gallic acid, wheat bran, phenolic compounds. Okay, cysteine, lipoic acid, gallic acid, wheat bran, and phenolic compounds. These are anti-mutagens. Most of the books do not mention the name of anti-mutagens. What are anti-mutagens? Any substances or any agent that decreases the spontaneous or induced mutation that will otherwise be caused by mutagens. What are mutagens? Mutagens are substances which have capacity to produce mutation. Okay, what are the examples of anti-mutagen? Cysteine, lipoic acid, gallic acid, wheat bran, and certain phenolic compound. Phenolic compound are found in plants. Gallic acid is present in strawberry and other fruits. Okay, so these are the examples of anti-mutagen. There is no other way rather than mugging this. Let's see the next answer. What is the role of low dose aspirin in ischemic heart disease? Okay, what is the role of aspirin in ischemic heart disease? Now there are two mechanisms of aspirin. Okay, there are two mechanisms of aspirin for the prevention of ischemic heart disease. First is what is happening in platelets and the mechanism which is mentioned by most of the books. So what is happening in the platelets? The arachidonic acid normally gets converted to PGG2. So prostaglandin synthesis is happening after this. Okay, but this first step, that is conversion of arachidonic acid to PGG2 with the help of COX-1, that is cyclooxygenase 1, this enzyme is inhibited by aspirin by acetylation of serine residue of the COX-1. Aspirin causes acetylation of cyclooxygenase 1 enzyme and makes this enzyme ineffective, so there is no thromboxane A2 synthesis, which has a potent vasoconstriction and platelet aggregation activity. So this is the first mechanism which is occurring in the platelet. Aspirin acetylates serine residue of cyclooxygenase enzyme. So it stops the synthesis of thromboxane A2, which has a potent vasoconstriction and platelet aggregation effect. Okay, this inhibition of aspirin by COX-1 is an irreversible inhibition. Okay, so decreased synthesis of thromboxane A2 leads to decreased vasoconstriction and decreased platelet aggregation. So decreased thromboxane A2 means decreased vasoconstriction and decreased platelet aggregation. So this is the first way by which aspirin is helpful in ischemic heart disease. Okay, inhibition of COX-1. Inhibition of COX-1, decreased synthesis of thromboxane A2, that means decreased vasoconstriction and decrease platelet aggregation. What is the second mechanism? Second mechanism is occurring in vasculature. Okay, here aspirin produces a compound which is called as ATL, aspirin triggered lipoxin. Okay, so what is happening in vasculature? Arachidonic acid is converted to an intermediate, that is 15R-HETE, that is 15R hydroxy eicosapentaenoic acid, okay, 15R-HETE. Here aspirin acetylates COX-2, okay, it acetylates COX-2. There is a production of this intermediate which is acted upon by 5-lipoxygenase and produces 15-EP lipoxin A4. This is called as aspirin triggered lipoxin A4 or ATL. What is the role of lipoxin A4? It has a potent anti-inflammatory effect. ATL, aspirin triggered lipoxin A4. It has a potent anti-inflammatory effect, and you know the use of anti-inflammatory effect. So that is another mechanism of aspirin in vasculature, production of ATL. Okay, so inflammation has a role in ischemic heart disease. Inflammation has a role for development of ischemic heart disease, so that inflammation itself is inhibited by this mechanism. Aspirin produces its own lipoxin A4, so that is why name is aspirin triggered lipoxin A4. So what is happening in vasculature, what is happening in platelets, we have seen previously. What is happening in vasculature? Aspirin acetylate vascular COX-2 and produces its own lipoxin A4, that is called aspirin triggered lipoxin A4. More amount of lipoxin A4 produced, that means more amount of anti-inflammatory effects. Okay, so aspirin causes acetylation of vascular COX-2, so there is redirects the activity of COX-2 from generating the prostaglandin to thromboxane to produce the intermediates, that is 15R-HETE, okay, and ultimately 15-EP lipoxin A4. This is called aspirin triggered lipoxin A4 ATL. This is another effect or another action of aspirin. That is the role of aspirin or low dose aspirin in ischemic heart disease. You have to mention two, not just a one. This aspirin has a role in platelet, what is happening in platelet, okay, and what is happening in vasculature. Okay, so this is the mechanism of aspirin in ischemic heart disease. Okay, next question is, that is the last one, define zymogens and give two example. Very straightforward question. Zymogens, what is define? 1 mark, examples 1 mark. Okay, example 1 mark, that is half mark for two, half mark for each. Okay, so what is zymogen? Inactive precursor of enzyme. Okay, inactive precursor of enzyme is called as zymogen, and you know the precursor, you know the examples like chymotrypsinogen, trypsinogen, procarboxypeptidase, proelastase, which are used during the protein digestion. So chymotrypsinogen is converted to chymotrypsin, trypsinogen to trypsin, procarboxypeptidase to carboxypeptidase, proelastase to elastase. Okay, so these are inactive precursor of enzyme. These are called as zymogen. They are produced as inactive precursor so that they are protected from the acidic environment. Okay, so these are the example. So thank you.